So that's me, Jason Klotzer. Says there I'm a customer engineer. More specifically, I love talking to folks about medical imaging, okay, whether it be radiology, cardiology, pathology, anything that can make an impact within that space. Medical imaging data, I would say classically has been one of the transformative sort of technologies within the sort of clinical care space. But it is a continuum, of course, right? So not only just in the provider space, being able to determine, what is actually going on within that patient's body, but also to be able to follow up on care, clinical trials. I'd say the whole data science space in general, when you look at FDA cleared, regulated models, vast majority of it is medical imaging based data. And a lot of that speaks to the complexity of the data, typically referred to as sort of unstructured data within a world of structured data. We all know that clinical data, saying it's structured is really a nice thing to say. So a lot of this is about the transformation in terms of where we are, in terms of classic sort of medical imaging applications, but also how can we get to the next level? That's really what we want to talk about. So that's me. Get the QR code. We don't have time for Q&A really, so I'd love to speak to you afterwards about any of these things, whether it be during this sort of session or at a follow-up. Rough agenda, I did a T-minus thing because these are massive lightning talks. So 50 minutes I've got, going to talk about the space, going to talk about, where are those improvement opportunities. I'm going to talk about what the accelerators are that we have in Google Cloud right now. And then, of course, no questions, sorry about that. But I'm going to have a quick demo on pathology, which is one of the sort of applications within the medical imaging space. So in terms of transformative sort of aspects of healthcare, I don't think anyone will argue that medical imaging is really one of those things that kind of is just obvious right now in terms of leveraging it for oncology care. But when we kind of look at it and we, kind of look at it outwardly, it's really impactful in many stages of the whole patient care sort of spectrum. In terms of early detection, when we're talking about, unfortunately, very prolific sort of areas right now, breast cancer, colon, lung, et cetera. Early detection is something that's super important, which I think we've all spoken about. Staging, if unfortunately it's found, it's present. Treatment planning when it comes to radiation, when it comes to surgery, and other different sort of methods. Guidance, effectiveness, in terms of actual treatment. And then lastly, of course, once the patient is over that hurdle, when they're truly not ever over that hurdle, really, how are they doing? How is this progressing? So many different sort of aspects of the clinical care kind of continuum is affected by medical imaging data. So this is something I want to spend a moment on, okay? So Forbes did both of these studies, but these numbers are kind of well known in the industry right now. 90% of the healthcare data is medical imaging data, okay? Which is, you look at that number and you're like, okay, well, images are big, right? So of course, it's going to be medical imaging data. The interesting thing about it is that over time, the quality of imaging data has just gotten to such a point that you can find things within imaging data that you couldn't have found 2 years ago, 3 years ago, 5 years ago. And that's really important to think about because that also affects, of course, your data size and what can be done within your downstream applications. Obviously, I'm going to start talking about analytics and AI and all the really interesting things you can do with that data, but I think just being cognizant of the fact that the data is so large to begin with, you have to take specific approaches to handling that. Very similar to how we talk about structured, unstructured in clinical data space. So that number, 90%, is probably a low ball at this point, considering the fact that we have pathology turning into digitized pathology at a rampant sort of click. We have, massive amounts of thin slice CT, we were talking about photon counting CT. So there are, tomosynthesis when it comes to mammography. The data is just expanding at a rapid pace within the healthcare organizations and across the world. The 90% on the right-hand side there is a really interesting one. So there's a massive amount of data out there, but this is saying that a patient comes in, diagnosis occurs, imaging, based on imaging and diagnostic interpretation, but then that data is no longer used. That's just crazy, honestly. The fact that that data is no longer being used at that point, I mean, one of the earlier sections was talking about, memes with cats and things like that. I mean, in a similar light, these images can represent so much data, okay? Whether it's, anatomy or, progression of, tumor, even, when we think about like all the characteristics of the patient and cohort, this information is so useful. We need to together change that number. We need to make sure that this data is leveraged to the point where we can actually make a difference. And that is really what a lot of providers, a lot of med tech, pharmaceutical through clinical trials, et cetera, organizations are trying to do. They are trying to leverage this data. And that's something we at Google Cloud, of course, and Google broadly are trying to help you with, right? We're trying to allow you to make sense of that data and be able to use it for its real potential, which is the original Forbes article there if you want to look into it. So this is one example of where imaging data is going to be leveraged. This is your sort of classic radiology interpretation workflow. I don't talk about the complexities of billing and claims and things like that because that's a whole other domain that, I would need to have like a 3-hour section to talk about that. But in terms of actual patient care and treatment, we typically talk about the initial consult, what's going on, do you have a genetic predisposition, is there something occurring in your life right now that would kind of cause this moment? If imaging needs to be done, you'll typically have, acquisition parameters configured, whether it be X-ray, CT, PET CT, et cetera, MR, ultrasound, whatever it might be. And as you can see, the details get pretty vast in terms of each one of these steps. Each one of these stages are things that could benefit from improvement. And that benefit from improvement typically comes through the lens of what has occurred in the past, what has been successful, what has not maybe been successful, and how can we learn from all this information. But the true situation is, back to that 97% number, is that a lot of this data is not leveraged. So if you're going to take one thing away from this slide, it is that there are so many areas for opportunity on improvement in a workflow such as radiology interpretation. So what are we as Google and Google Cloud doing to try to help within this area? Feel free to take a picture of this slide. Each one of those QR codes will hopefully work and point you to something within this space. But we have 3 sort of pillars with respect to medical imaging data specifically. First is what we call our medical imaging suite solution. And this solution is really around attempting to standardize that data. Traditionally in radiology, we're talking about DICOM, but it's not always just DICOM, so multimodal comes into account most of the time. If you want to have any useful data within radiology, you're typically going to be looking at minimally the imaging data and the diagnostic interpretation, which is typically a big blob of text, sometimes structured if you're lucky. But being able to make sense of that data is something we do a fairly good job at. Obviously I'm underselling that. But that's something that Medical Imaging Suite is really great at. And we take a very specific focus on medical imaging, clinical data specifically, and how to leverage that. The next category there is our foundation models. You'll see a bunch of names there that you're probably familiar with. Of course, our sort of lighthouse, multimodal model Gemini is there. But we have so many foundation models that can act as accelerators for your interests within, accelerating patient diagnostics, accelerating, maybe even turnaround times or, operational improvements. So we've got chest X-ray, CT, which is the latest that we actually just released, I think, last week, the CT foundation model. Dermatology, something I'm going to demo very briefly, pathology, histopathology specifically. My ask is just look them up, see if they can help you. Again, these act as accelerators for what you're attempting to do. We also have several partners around the room who also have applications of these models that you can kind of see their demos and see how they're, reaching the, the sort of providers and care continuum partners as well. And then lastly, of course, as I'm talking about partners, our partner ecosystem is so incredibly powerful, okay? Talk to our partners, have us introduce you to partners, let's do something together. This, cancer, oncology is 100% team effort. Nobody's gonna go it alone, so let's talk to our partners within the space. This is just one example, as you can see. Samir Sethi, the sort of guru of data science and data analysis and analytics within Hackensack Meridian Health. Hackensack implemented medical imaging suite, I think it was 2 years ago now at this point. But this pattern is very, very reproducible in the sense that it was mainly around aggregating data, being able to understand what the data is, being able to search the data, being able to find, consistencies or inconsistencies in the data, and this is all information that's necessary if you're ever going to want to build a high accuracy model or even an analytics pipeline to improve operations, et cetera. So great example at Hackensack. And then there is a quick demo here. I asked the folks to speed this one up, but effectively what this is doing is taking DICOM WSI histopathology-based data, which if any of you have ever worked with it, I'm sure there are some pathologists in the audience. This data is massive, okay? We're talking about tens of thousands of pixel by tens of thousands of pixel. And this is an example of how we took a foundation model, which is our histopathology model. We uptrained it on being able to see similarities within the slides. And we effectively created an example pipeline for doing image-based similarity in real time within histopathology slides. And this is a specific example for breast cancer within the lymph node. But as you can see here, we're selecting a portion within the massive slide. You might not be able to see this, but in terms of actual size and actual pixel ratio, we're talking about a massive slide here. So what it's doing is in real time, it's actually calculating a region of interest or multiple regions of interest within the whole slide that fit the initial criteria in terms of cancer within the lymph node. And as you can see here, it was completed within a couple of seconds. Just these kind of things can massively accelerate a pathologist's workflow. Whether it's just things like cell counting or similarity detection, helping accelerate in terms of staging or various other tasks that may not be interesting but massively affect clinical care. That's one great example. And that's what I've got for you. 1 minute to spare. So please, if you'd like to have a follow-up conversation, find me in the audience, find me after the fact, but it's been a pleasure. So.
Hi, my name is Vincent Beltrani. I am a healthcare and life science specialist at Google Cloud. And in a previous life, I was a computational chemist, so I am really enjoying being here at the conference. I'm joined with Dr. Petrina Kamya, the global head of AI solutions. and AI platforms at Insilico Medicine. So what I want to talk to you today is about AlphaFold and AI-powered drug discovery with AlphaFold. But before I get there, I want to talk about the first disease that we understood its molecular basis for. That first disease is sickle cell anemia. And that's a disease of red blood cells. In fact, it's a disease of the hemoglobin protein itself. Sometime in the late 1940s, Linus Pauling and his team at Caltech pinpointed the exact molecular cause of sickle cell anemia. And believe it or not, it comes down to a substitution of one single amino acid in the hemoglobin protein. So the normal hemoglobin protein has a structure that makes it very efficient at capturing oxygen and being packaged into a red blood cell. In the case of sickle cell anemia, the distorted hemoglobin protein instead is much less efficient at capturing oxygen, and inside the red blood cells, that hemoglobin polymerizes, and it creates that sickle cell red blood cell shape, which makes it very difficult to transport oxygen around the body, and it leads to things like anemia. The reason I use that example, one is because it's very, very interesting, and then the other one is that it really highlights the kind of important fact that a protein's structure determines the protein's function. Everything in your body, all of those biological processes involve proteins. And if you want to be able to understand disease, if you want to be able to create therapies and create treatments and drugs, you need to understand the protein or the structures of those proteins that are involved in those biological processes. So how do you determine the shape of a protein? Well, we have a number of experimental techniques to determine the shapes of proteins. We have X-ray crystallography, we have NMR, we have cryo-EM. Those are amazing techniques, but they're expensive, they take a lot of time, and they're constrained. I need to be able to isolate my protein. I might have to crystallize my protein. I might need to purify my protein. At the end of the day, the protein universe probably contains hundreds of millions of possible proteins, and we really only have experimental structures for about 200, 300,000 of them. So it's been a long-standing goal in computational science to develop a method to be able to predict the 3D structures of proteins directly from their amino acid sequences without needing to run either expensive experiments, timely experiments, or for some types of proteins, impossible experiments. I will kind of fast forward the history of the protein folding problem. People took many different approaches, but I'll start in 2017 when Google DeepMind released a seminal paper called Attention is All You Need. In that paper, DeepMind introduced the world to the transformer architecture and the idea of self-attention. 2 years later, 2019, 2020, building on top of that work, Google DeepMind releases the AlphaFold protein prediction system to the world. The first protein structure prediction system that can generate protein predictions that are close to or within experimental accuracy. So what I'm showing here on the screen are just two proteins that have been folded by the AlphaFold system. In both cases, the root mean squared deviation is less than 1.5 angstrom, meaning that each atom in these compounds or in these proteins is within 1.5 angstrom from its experimental value. What that means for drug discovery is that these predicted structures are biologically relevant. That means that I can use these structures as a starting point for my drug discovery efforts. That might be trying to find hotspots on my protein. That might be identifying potential binding sites. And that might be for hit finding in my virtual screens. So it really is a revolutionary time in drug discovery with a combination of deep neural network, generative AI, tools like AlphaFold, and the computational power that you can get on your cloud provider like Google Cloud. So now I want to introduce Dr. Petrina Kamya to talk about their work using AlphaFold in their drug discovery efforts at Insilico Medicine. Thank you.
Thank you. Thank you, It's on. Thank you, Vincent, and thank you, Google, for inviting Insilico Medicine here to talk about our, I believe this was the first paper that was published that experimentally validated an output that came out of AlphaFold 2. So just a brief history or background of Insilico Medicine, why we here, why is our story relevant to Google's story with AlphaFold 2. So we are a tech bio company. We basically have two pillars. We develop technology that we leverage heavily to accelerate the process of drug discovery and development throughout the entire value chain of that process. And we also develop our own pipeline of therapeutics. So being one of the pioneers in this space, we've been around for about 10 years. We've built out this AI drug discovery platform called Pharma.ai. This is where I sit. I'm global head of this platform. And we've also built out an impressive pipeline of therapeutics. We currently have 31 internal programs. One is in phase 2 clinical trials. We just got a positive readout from our phase 2A clinical trial. We have 5 programs in phase 1 and over 10 programs in IND-enabling studies. And having moved these programs so quickly into the clinic and so fast throughout the entire process is basically because we've heavily leveraged AI and AI-based technologies. And the main thesis of the company is to go after the traditional approach of drug discovery and development, which typically takes a long time and costs a lot of money. Our approach is to shorten the time it takes, leveraging as much technology as possible so that we can bring drugs to patients faster. And we've demonstrated that this is possible through our internal pipeline. So this is what our current platform consists of. We have basically 4 pillars: biology, chemistry, and medicine, where under each of these different pillars, we leverage many different deep learning technologies to accelerate the process from target discovery all the way up to going into the clinic. And this is just a snapshot of some of the programs that we have demonstrated the value of leveraging this type of technology looks like. So being a tech-first company and having this thesis where we really wanted to shorten the amount of time that it takes from idea to the clinic, when AlphaFold 2 came out, we were very, very intrigued by this technology because being a computational chemist myself, I fully understood the difficulty it was when it comes to understanding the relationship between a protein structure and its function, and then leveraging that structure using models to design molecules and therapeutics that would target that structure. And we are a company that develops tech, yes, but if there's a technology that's already out there, why wouldn't we leverage that to see if it can help us accelerate our programs and, live according to our thesis? So this is a paper that we published about 2 years ago. And like I said, we were one of the first people to try to take AlphaFold 2 derived structures and experimentally validate them. So we took a disease that had high unmet need, hepatocellular carcinoma. It's a liver cancer. It's the 6th most common cancer in the world. Most of the cases of liver cancer are hepatocellular carcinoma. Major risk factors include those that I've shown above. I'm running out of time, so I'm going to move a little faster. Standard of care is not that great. Basically just increases the overall survival rate of patients with this disease by about 19 months. So this is a disease that was of interest, and we can leverage one of our technologies to try and identify novel dark targets, so targets that don't have a crystal structure or a known structure, to see if we can use an AlphaFold 2 derived structure and design molecules that target this particular protein with the hope of generating therapeutics that could help treat the disease. I'm sorry, my slides have been Googleified. So they look a little bit different, but anyway. So basically, what we wanted to do is identify a combination therapy partner that will increase the overall survival rate in the frontline setting. And we identified a target, CDK20, which is called cyclin-dependent kinase number 20, which has been shown to inhibit T cells and support tumor growth in patients with hepatocellular cancer. So we found this connection in our platform, PandaOmics. And it therefore would serve as a potential target in this particular disease and help with this high unmet need. So there we had our starting point. We had a disease of interest, and we had identified a target that had no known crystal structure. So we therefore took the following steps. First, we found the structure of CDK20 in AlphaFold 2. We identified key features that we could leverage for the design of selective CDK20 small molecule inhibitors. We then leveraged our other platform, which is a multi-agent reinforcement system, heavily relies on generative AI to design and optimize small molecules that could be potential therapeutics. And then we synthesized and tested those molecules to see whether or not leveraging an AlphaFold 2 structure, dark structure, can result in the design of molecules that are actually experimentally validated. So how did we do? In only 30 days, we went from identifying this target to having a small molecule therapeutic that has been validated experimentally for, as a potential therapeutic against hepatocellular carcinoma. This is the structure of the molecule, so that people can see that it's actually real. And we were able to do this in such a short period of time by leveraging AlphaFold 2. Another very key important fact is that this was the very first time that someone actually had done this, taken a protein structure that had no previous known folded structure and actually generated molecules that could be potential therapeutics in a very short period of time. And I guess my conclusion is to say that machine learning tools have come a long way over the last 10 years. And thanks to companies like DeepMind, we also have Recursion here, we have Ginkgo Works, Insilico Medicine. I do believe that as pioneers in this space, we're really showing the potential that machine learning tools can have and the impact that they can have on shortening that time it takes to get drugs to the clinic, to patients, at a significantly smaller cost than what is currently the status quo. I have 3 minutes to spare. I've never really, this is a new experience for me, but thank you very much.
Good morning. I'm Jing. I work for Illumina. Sumeet and I, let's take one more look at the photos. Sumeet and I will introduce the type of work that Illumina has been doing and in collaboration with Google to help advance precision medicine. 20 years ago, Illumina helped build the foundation of genomics with its groundbreaking sequencing technology. And for 20 years, we continue to drive innovation to lower sequencing cost and to enable a broad range of life science applications using our technology. Our mission is to improve human health by unlocking the power of the genome. Cancer is a heterogeneous disease. Many talks today also reviewed some of the challenges. Each cancer case involves different types and different numbers of genetic mutations. It is very challenging to locate the driver mutations among the millions, possibly millions of genetic variants in a cancer genome. It is even more challenging to find good ways to synergize complex genomic information with human medical expertise. And AI can help us to separate signal from noise in this case. Possibly more importantly, AI allows for incorporation of advanced science, very complex information into clinical routine. And Illumina leads AI for genomics for many reasons. To give a few examples, we have access to the largest data sets, the most diverse data sets. Some of these data sets are unique, some are proprietary to Illumina. Also, we excel in feature engineering because we understand Illumina sequencing technology natively. So all of our AI components are enhanced with sequence read-based features. Illumina has a dedicated 40-people AI team. This team has, on average, more than one doctoral degree per person, a very nerdy team. They publish on a regular basis on top-tier journals on Science, Nature, Cell, first author and last author publications. Their amazing work made it possible for Illumina today to have some of the most advanced AI capabilities in genomics. We make this statement seriously, not only with confidence, but with evidence, because the quality of Illumina AI publications today is unmatched among all technology providers in genomics. And these proprietary AI components are built into every step of our data analysis solution and our data analysis pipeline. The fact is that our goal is beyond building AI ourselves. We are a technology provider. We support large population-scale genomic initiatives worldwide to generate data for medical research. We also supply AI and advanced algorithms to our collaborators for their research work. The way we look at it, Illumina's industry-leading sequencing technology, our large, diverse data collections, and our advanced software solutions together support a very powerful iterative process for genomic communities to advance AI together. Specifically for precision oncology, Illumina's Dragon supports secondary analysis for all common oncology applications, for therapy selection, for treatment monitoring, and for hereditary risk identification. Possibly more interesting to this crowd, our Dragon also supports data analysis for all advanced cancer research in the areas of epigenetics, pharmacogenomics, single-cell sequencing, proteomics, neoantigen identification, and more. Illumina Dragon is widely recognized as the most accurate secondary analysis today in genomics. It has won first place for best overall accuracy in the Precision FDA Truth Challenge 4 years in a row. AI, along with advanced algorithms, allows us to maximize the information and the value we can extract out from the genome. And our tertiary interpretation software solution, Illumina Connected Insights, built specifically for precision oncology, brings meaning to the genetic information. This software solution annotates variants with 55 data sources, including premium content from commercial knowledge bases such as CKB and UncleKB, all free of charge to our software users. Now, I'm sure many of us can appreciate the difficulty in AI with variant annotation, because when we build AI models with only available data sources, AI won't be able to tell us anything more than what we don't already know. So the Illumina team solved this problem by augmenting the training data sets with non-human genomic information. Illumina sequenced a large number of primate samples across multiple primate species, and we built AI models to predict the functional impact of human variants within a cancer genome based on cross-species sequence conservation and protein folding. This innovation was published on Science and was selected as the cover for the issue. Since this publication, many studies have been performed to validate Illumina's Primate AI 3D for its accuracy and its coverage. And in all of these studies, some are held in collaboration with Illumina, many are independent. Illumina's Primate AI 3D outperforms all other existing functional predictors. The Illumina AI team pushed their innovation even further and built 5 additional AI capabilities for the non-coding regions of the genome. We heard from the earlier presentations today. Now we're looking at the whole genome to find answers for cancer. Take, for example, Splice AI. Illumina team utilizes 10,000 nucleotides along with multiple exon sequence to predict the splicing events, compared to previous algorithms that are based only on motif recognition. Splice AI demonstrated superior performance. In some data sets, we see around 18% of accuracy by the motif recognition algorithms, and Splice AI, in contrast, demonstrated over 96% accuracy. And it was recommended by guideline publications for predicting oncogenicity. Altogether, the 5 AI for the non-coding region, we estimate can help increase diagnostic yield by 15 to 39%, depending on the cohort used for validation. The way we look at it, right, how data solutions should work for precision oncology, we believe high-quality sequencing readout coming off Illumina sequencers is translated into fully featured genome by our secondary analysis solution. That is, the entire genome, every variant is fully featured, and then annotated with comprehensive clinical insights by our tertiary interpretation solution. Now, AI in this time of information gives us the opportunity to present the whole genome as a platform of information where complete molecular assessments can be performed throughout the patient journey. Now, one example is hematological cancer. This is a cancer that's traditionally tested by multiple methods in a sequential manner. In fact, on average, most patients will be tested 4 to 5 times at the time of diagnosis, then another 4 to 5 times during the course of treatment. The reason so many methods are used is because different types of variants need different types of identification methodology. Each method is simple. Now, they include qPCR, targeted gene panel sequencing, FISH karyotyping, genome mapping, microarray, and so on. Each method is simple. However, collectively, they create a diagnostic odyssey that's financially and emotionally burdensome, not to mention the inconsistency, right? The different type of medical expertise coming together to agree or disagree while the patient is waiting. The Illumina approach is one single test that identifies all variants of all types that has been published by leading hospitals and laboratories worldwide. On the screen is an example of AML, acute myeloid leukemia. AI, in this case, followed WHO 2022 diagnostic guideline. Within hours after sequencing, automatically prepared a comprehensive diagnostic and prognostic report with all the information ever needed. Now, I want to mention that when we build AI for precision oncology, we only build explainable AI. That is, information used by AI, decision trees followed by our algorithms, sources of information, even the genomic context of key driver mutations, the sequence contact, are clearly displayed along with the AI recommendations for human inspection, and also collect human corrections for AI to self-learn. As we build solutions for clinical routine, we understand that in order to conquer cancer, we must look into large cohorts. We must look into phenomics and environment and returning value across diverse populations. Illumina has a long history of supporting population-scale genomic initiatives across the globe. These are some recent programs. In each case, hundreds of thousands of participants, patients and healthy individuals are sequenced, many from diverse populations, and advanced research is performed using our AI and software solutions. Now, the data generated is fed back to improve AI and data solutions. To date, on the global scale, we have analyzed close to 5 million genomes. We don't have time to go into greater details today. I look forward to our offline discussions. And Sumeet will introduce our collaboration with Google. We hope for opportunities to work with you and support your research work.
Thank you, Jing, for your partnership and for being here today. My name is Sumeet Ranu, and I'm a customer engineer at Google Cloud, working with our top healthcare and life sciences customers like Illumina to solve healthcare problems with the power of cloud computing technologies. Today, I'm very excited and proud to announce the availability of Illumina's Dragon on Google Cloud. This is a solution that's been a labor of love over the past couple of years, and a lot of people were involved in making this happen. This brings together Illumina's software, the Evident hardware, and then Google's cloud platform to allow our customers to run Dragon workload secondary analysis on Google Cloud. So some examples of folks that would benefit from this would be researchers and bioinformaticians that might be running Dragons on-prem today. And they could use this to either move some of the workloads to the cloud where their data might reside, or they could go ahead and burst and scale up using Cloud Dragons. So I want to go over a few benefits and features that make this solution stand out. First and foremost, our solution uses state-of-the-art field programmable gate arrays. These are accelerators that greatly enhance the speed and performance of the computations that are required. It takes a lot of horsepower to sequence a genome. So these FPGAs were procured and installed by Evident and Google in a data center. And we have a software abstraction layer that we built that lets you interact with those through your Google Cloud resources. And that brings me to my second point. This solution allows you to leverage your already existing Google Cloud resources as well. So for example, we have a lot of customers that are storing genomics data in Google Cloud storage. This solution allows you to plug and play, take that input data, and then put the outputs in the same location. It also works with the rest of your Google Cloud ecosystem, your organizations, the way you have everything set up. So it's fairly seamless. And last but not least, this solution also allows customers to tailor the workflow to what they need. So for example, a bioinformatician might have a very specific workload workflow they created that works for them for on-prem. And in that case, they might want to take our bare bones code and go ahead and integrate that into their solution. So that's just one example. Other options include using our batch integration code that we developed, so we can give you access to that so you can run that. And then the third option is the Cromwell integration. So a lot of bioinformaticians and researchers use Cromwell today, and that integrates with this solution as well. We are also working on a few other popular tools, so that's on our roadmap. And if there's one that isn't listed on here that you want to see, please let me know. We can add that too. Now let's dive into the architecture a little bit of what this solution looks like at a very high level. So the entry point into the solution is through Google Cloud Batch. Batch is an orchestration engine that lets you schedule and execute tasks at a low level. And behind the scenes, Batch uses Google Compute Engine, our virtual machines in the cloud. Lets you execute one task at a time, and once the task is done, it does the cleanup for you, which is the nice part about Batch. So Batch is the entry point into the solution. When you set this up in your environment, you're going to set up a configuration that tells Batch exactly where to find your input data, where to store your output data, and then also Dragon-specific configuration. So that includes your Illumina Dragon license key, if you have one, and also the Dragon command line options you want to run depending on the samples you're running through. So once you set that up in Batch and configure that task to run, it sends a signal to the FPGAs that are in a data center. So the FPGAs are abstracted away from the customer, but it's important to know how this works. These FPGAs are managed by Evident and Google, and they're located in a colo that's close to our Iowa data center, and there's a dedicated interconnect between those two. So all the data is transferred securely and very fast. That's where the majority of the compute happens, the Dragon compute that Illumina developed. So once the FPGAs are done running the Dragon analysis, it creates these output files. And it stores those output files back into Google Cloud Storage, which is our object storage solution. So you can have all your files all in one place and ready to go. And then this is where our customers can use it for downstream processing. Let's talk a little bit about the Dragon piece here. So you might already have an agreement with Illumina or Dragon licenses that you're using. And the great thing about this model is it's BYOL, bring your own license. You can use that and you're ready to go. It will depend on your agreement with Illumina. Definitely reach out to them, but it's ready to go. And then last but not least, any data that you store as a result of this process, you're able to use all of our other Google Cloud services with it. So you have logging, monitoring. You can also use analytics on top of it for whatever you need to do with it. So that's a little bit about the solution today. We have a lot of customers using the solution already in production, and we're looking for more. So if you're interested, please reach out to me. Our customers are running thousands of samples per month in this solution, and we're growing at a very fast rate. So if you're interested in learning more, or if you want to just test the solution out, please find me during lunch or afterwards, or you can reach out to me at dragon@google.com. Thank you for your time today.